Today our topic is bacteriophage. Viruses that attack the bacteria were observed by Edward and Hell in 1915 and 1917. They observed the growth cultures of certain interstitial bacteria could be dissolved by the addition of the bacteria-free filtrate obtained from the sewage. Let us speak about the brief history. It was discovered in 1915 or 1917 basis on the clearing in the bacterial lawn and the propagation as the infectious agents. While speaking about the ideas, the nature of a virus and the phage therapy, using the phages as an agent to combat the bacterial infection. Modern era of phage research in 1940, the nature of a gene and using the phages as an experimental model system. Extra results like the nature of a gene and the expression of a genes and the expression regulation, development of the methods of the recombinant DNA. Bacteriophage, let us see the definition of it. They are obligate intracellular parasites that multiply inside the bacteria by making the use of some or all of the host biosynthetic machinery. Their significance are they are the models for the animal cell viruses. They can be used in the gene transfer in the bacteria. While speaking about the medical application, the identification of a bacteria like phage shaping and they are also used in the treatment of the prophylaxis. Bacteriophages are the virus that can infect and destroy the bacteria. They have been referred to as the bacterial parasite, with each phage type depending on the single strain of the bacteria to act as a host. Bacteriophage. Like most viruses, bacteriophages typically carry only the genetic information needed for the replication of the nucleic acid and the synthesis of the protein code. They require the precursor, energy generation, and the ribosome supplied by the bacteria host cell. Bacteriophage classification. At present, over 5,000 bacteriophages have been studied by the electron microscopy and can be divided into 13 virus family. ICTV designated phage families: gamma, T4, T7, PRD1, X174, M13, MS2, 6, PM2 and L2. While speaking about the family, let us see the family prototypes and the characteristics. Cypheviridae, this is the gamma type. Their characteristics are dsDNA. They are long, non-contractile tails. Myoviridae, T4, they have dsDNA on the contractile tails. Podoviridae, they are T7, dsDNA, short and the stubby tails. Tectiviridae, they are PRD1, dsDNA, linear, 5' proteins and the internal membranes. Microviridae, X174, ssDNA, circular and icosahedral. Rhinoviridae, M13, ssDNA and the filamentous. Labioviridae, MS2, VB, ssDNA, small icosahedral. Cystoviridae, they comes under the 5-6 dsDNA, segmented enveloped. Corticoviridae, PM2 dsDNA, circular internal membrane. Plasmaviridae, L2 dsDNA, circular and enveloped. Bacteriophages make up the diverse group of viruses, some of which have been complex structures, including the double-stranded DNA. Also known simply as a phage, a virus that attacks and infects the bacteria. The infection may or may not lead to the death of the bacterium, depending on the phage and sometimes on the conditions. Each bacteriophage is specific to one form of the bacterium. While speaking about the composition and the structure, the composition they have nucleic acid depends upon the genomic size and the modified bases. They also have the protein, which is used for the protection and infection. This structure is the T4. Size, head or capsid and tail will be present. Virulent versus potent cells. Virulent phages, they do not integrate the genetic material into the host cell chromosome and usually kill the host cell, that is the lytic infection. Temperate phages, they may integrate into the host DNA causing the lysogeny. Bacteriophage virulence factors carried on the phage. Temperate phage can go through one of two life cycles upon entering the host cell. One is lytic one. This is when the growth results in the lysis of the host and the release of the progeny phage. Lysogeny, this is when growth results in the integration of the phage DNA into the host chromosome or the stable replication as a plasmid. Most of the gene products of the lysogenic phage remains dormant until it is induced to enter the lytic cycle. Lysogenic conversion. Some lysogenic phage carry the genes that can enhance the virulence of the bacterial host. For example, some phages carry the genes that encode the toxin. These genes, once integrated into the bacterial chromosome, can cause the harmless bacteria to release the potent toxin that can cause the disease. Bacteriophages are used for the cloning the foreign genes. Among the other applications, proteins or peptides are fused to the capsid of the phage. The combination of the phage and the peptide is known as a fusion protein. Bacteriophage attaches to the host bacterial cell and injects the DNA. Bacterium, phage, gene product and the phenotype. Let us see the virulence factors carried by the phage. Vibrio cholerae, their phage is CTX phage, cholera toxin, that comes under the phenotype cholera. Escherichia coli, lambda phage, Shigella toxin, hemorrhagic diarrhea. Clostridium botulinism, clostridial phages, botulinism toxin and the botulism. They are used in the phenotype of food poisoning. Corn bacterium, diphtheria. Corn phage beta, diphtheria toxin, diphtheria. Streptococcus pyogenes, their phage is T12. Their gene product is erythrogenic toxin and they are caused by scarlet fever. Lysogenic conversion. In some interaction between the lysogenic phages and the bacteria, the lysogenic conversion may occur. It is when a temperate phage induces a change in the phenotype of the bacteria infected that is not the part of a usual phage cycle. Changes can often involve the external membrane of the cell by making it impervious to the other phages or even by increasing the pathogenic capability of the bacteria for the host. Assay for the lytic phage. Plague assay. Their method is plague forming unit. They measure the infectious particles. The DNA of the temperate phage enters into the bacterial host cell. The phage DNA may become integrated with the host cell DNA as a prophage. When the prophage is induced and leaves the bacterial chromosome, it may carry along the piece of the bacterial DNA in place of the phage DNA. The phages that are replicated are the defective because they lack some viral genes that have been replaced by the bacterial DNA. Note that the phage head contains both the phage and the bacterial DNA. The defective phage DNA enters the new host cell but cannot cause the production of the new phage particles. Bacterial genes introduced into the new host cell or integrated into the DNA becomes a part of the bacterial chromosome and are replicated along with the rest of the bacterial DNA. While speaking about the examples of the bacteriophages, corn bacterium diphtheria produces the toxin of diphtheria only when it is infected by the phage beta. In this case, the gene that codes for the toxin is carried by the phage, not the bacteria. Vibrio cholerae is a non-toxic strain that can become the toxic, producing the cholera toxin when it is infected with the phage CTX. Clostridium botulinism causes the botulism. Streptococcus pyogenes causes the scarlet fever. They also causes the sugar toxin and the tenderness. Medical applications of the phages. I strongly believe phage could become an effective antibacterial tool, said by Carl Merrill, who is the chief of the laboratory of the biochemical genetics in the National Institute of the Mental Health. It might be the another string on the bow, such that when fail, here is something that has a chance of working, but it's not going to be panacea, said by the Joshua Liedenberg from the Rockefeller University. Model organisms. Fundamental problems are solved in the simplest and the most accessible system in which the problem can be addressed. This organism are called the model organism. Some important model organisms like the Escherichia coli and its phages, T phage and the phage lambda, Baker cyst, Saccharomyces cerevisiae, nematode, Caenorhabditis elegans, the fruit fly, Drosophila, house mouse, mus musculus. Features of the model system. The availability of the powerful tools of the traditional in the molecular genetics. The study of each model system attracted a critical mass of the investigators, like in the ideas, methods, tools and the strains could be shared. How to choose a model organism? It depends on what question is being asked. When studying the fundamental issues of the molecular biology, simpler unicellular organism or the virus that are convenient. For the developmental questions, more complicated organisms should be used. Bacteriophage, this is the type 1 virus. The simplest system, their genomes are replicated only after being injected into the host cell. The genomes can recombine during this infection. Each phage attaches to the specific cell surface molecule, usually a protein, and so only the cells bearing that receptor can be infected by the gene given phage. Two basic types, lytic phage. That is the example T phage. They infect the bacterial cell, DNA replication, code protein expression, host cell lysed to release the new phage. Temperate phage, phage lambda lysogeny. The phage genome integrated into the bacterial genome and replicated passively as a part of the host chromosome code protein. That is, the genes are not expressed. This phage is called the prophage. The daughter cells are lysogenes. The lysogenic state can switch to the lytic growth called the induction. Excision of the prophage DNA replication, code protein expression, and the lytic growth. Assays of the phage growth. Propagate phage by growth on the suitable bacterial host in the liquid culture. Quantify phage, plague assay. Propagate phage. They find the suitable host cell that supports the growth of the virus. The mixture of the viruses and the bacteria are filtered through the bacteria-proof filter. Quantify phage. Phages are mixed with and adsorbed to the bacterial cells. They give the mix. They add the dilution to the soft sugar, contain many uninfected bacterial cells. Poured on to hard agar base, incubated to allow the bacterial growth and the phage infection. As the virus replicates and are released, they spread and infect the nearby cells. The circle of death produces a hole or plague in the lawn of the living cell. These plagues can be easily seen and counted so that the number of viruses can be quantitated. The single-step growth curve. Latent period. The time lapse between the infection and the release of progeny. Burst size. The number of phage released. The single-step growth curve. It reveals the life cycle of the typical lytic phage. It reveals the length of time it takes a phage to undergo the 1/3 of the lytic growth and also the number of progeny phage produced per infected cell. Method. The phages were mixed with the bacterial cells for 10 minutes, long enough for the absorption but too short for the further infection. The mixture is diluted by the 10,000. Only those cells that bound the phage in the initial incubation will contribute to the infected population. Progeny phage produced from those infections will not find the host cell to infect. Incubate the dilution. At intervals, a sample can be removed from the mixture and the number of the free phage counted using the plague assay. Phage crosses and the complementation text. This is a mixed infection. A single cell is infected with two phage particles at once. It allows one to perform the phage processes. If two different mutants of the same phage co-infect a cell, the recombination can occur between the genomes. The frequency of this genetic exchange can be used to order the genes on the genome. It allows one to assign the mutation to the complementation group. If two different mutant phage co-infect the same cell and as a result each provides a function that the other was lacking, the two mutations must be in the different genes. Complementation groups. If not, the two mutations are likely coded in the same gene. Transcription and the recombination. During the infection, a phage might pick up a piece of the bacterial DNA. Mostly happens when the prophage excises from the bacterial chromosome.